Okay, let's move on to nervous system. As I've said, coordinates your behavior, information passes along the neuron cells, and their electric impulses, as mentioned before. These electric impulses go to your CNS, the central nervous system. Your CNS being your brain and your spinal cord, but we're talking mainly the brain. Let's not confuse ourselves. The CNS, as said here, brain and spinal cord, and it coordinates responses involving effectors. Effectors can be things like your muscle. That's an effector. So there are three types of neurons you need to know about. There is the sensory, the relay, and there's also the motor. The sensory, relay, and motor, it's kind of think of it as like a linking from A to B to C. It goes from sensory and then to relay and then to motor. But the steps regarding it is you have receptors, so things that detect the stimulus. So if there's a change in environment, receptors are going to detect it. So for example, touch. There's going to be receptors on your fingertips. You're going to feel this touch. Receptors detect the stimulus, carries the electrical impulses through the sensory neurons from the receptors to the CNS. This electrical impulse is going to go through the sensory neuron. Then it's going to go through the relay neuron through the CNS. And after you've gone through the CNS, through the relay neuron, this electrical impulse is going to continue down to the motor neuron and from the CNS, and this is going to be linked to the effector. The effector is then going to produce a response. And I'm going to show you an exam question later to show you kind of what I just said. But the steps essentially is just receptor, sensory neuron, relay neuron, which is through the CNS, motor neuron, effector, response. Step by step. I'm going to say that again because that's usually the easiest way people understand it. Response. So, no, sorry, not response. Sorry. Stimulus, sensory neuron, relay neuron through the CNS. So CNS, yeah. And then motor neuron, effector, response. That is step by step what is exactly is happening. So I'm going to show you an exam question on that. Okay. Oh, there is no exam. Wait, okay, where is it? Let me just go all the way back because I think this is a really good example here. So I did say we won't go through this, right? So it asks you to describe how the diagram brings about this reflex action when someone touches a hot pan. So when someone touches a hot pan, like you're going to detect this temperature. It's hot, so you're going to want to move this away. So when you detect it, receptor detects the stimulus. So the stimulus being the heat. So you see the first one here, stimulus, heat detected by temperature receptors in the skin. This electric impulse is going to then travel through the sensory neuron to the CNS. This is then going through the CNS, through relay neuron, and then to the effector through the motor neuron. So motor neuron and then effector. And then the muscle contracts, which is the response. So you have to be more specific when you're answering question through the response. Also, there's another point you're talking about chemical transmissions across the synapse. Don't stress too much about that point, but it's just something you need to know when you're answering the question. As I've said, between neurons, there are synapses, and this is how they link to each other. They have signal to each other. Between these synapses and these neurons, the transmission of signals is through chemical transmission. So that's just something to point out. But that's kind of the whole of the nervous system. If you have any questions on just the basics of it, please let me know. The next section for the eye is only in triple science. So if you're in double science, I guess have a break. Like in combined science, have a break. But the next part is triple science. So I'll just quickly explain this. For triple sciences, you need to know about the eye very specifically. So there are two things that you need to know about the accommodation of the eye. First one is looking at just different distances, looking at something close and looking at something far away. Then for adaptation to light is looking at something very bright compared to looking at something very dark, very dim light. So let's look at that. You need to know the structure of the eye, first of all. I'm not going to go through every single structure and every single function. It's in the table right there if you want to have a look, but you do need to know them well. So if you're looking at something near, and if you look at different distances, we have our ciliary muscles and our suspensory ligaments, and they work antagonistically. What I mean by antagonistically is that they work together, but opposite. You'll understand what I mean when I explain that. If you're looking at something near, your ciliary muscles are going to contract, your suspensory ligaments are going to loosen and the lens becomes shorter and thicker to see something near. So you can see how they work oppositely. Ciliary muscles contract, suspensory ligaments loosen, but they work together to allow the lens to become shorter and thicker. This allows us to see things near. Things that are far away, opposite. Ciliary muscles relax, suspensory ligaments are pulled tight, so contract essentially, and the lens is thinner and longer. So that's looking at things at different distances. Adaptation to light is when there's different light intensities, and this allows our pupils to dilate and constrict. So the reason is because if there is a very bright light, you don't want to damage your eye, so it constricts, makes it smaller, so not too much light is going in. And if it's very dim light, you're going to have these your pupil dilate, so that when it's larger, you get more light in, so you can see in darker light. So in this case, when there is a very low light intensity, the radial muscles contract and your circular muscles relax. Again, they work antagonistically to allow the pupil to dilate or constrict. And in bright light is the opposite. Radial muscles relax, circular muscles contract. So that's the eye. There are a few questions on here. However, I am wary that there are people who are doing combined sciences here as well. So if you are doing triple sciences, have a look at this after the seminar, after the, oh my God, hold on. Right. The light's off again. But yeah, if you guys have any questions on the eye, just look through it yourself after the thing. Okay, can you go back to the eye? Yes. What do you want me to look through in the eye more specifically? Yes. Okay. Yeah, no worries. Just wait, hold on. Give me a second, guys. The light is off. Hold on. Okay, sorry. Okay. Perfect example for light and dark right now for the eye. Okay, let's explain that. So I'm going to explain it again with the circular muscles, radial muscles stuff. Okay. I think everyone, kind of understood the near and far one. I feel like that one was, I think, explained more clearly than what I said to the light thing. So let's go back to the light. So when you're in very, very bright light, your eye is going to want to, your pupil, so basically the hole that allows light into your eye, is going to want to go smaller, constrict. They're going to want to go smaller because you don't want to damage the eye. Too much light in the eye is going to damage it, yeah? So they're going to constrict the pupil. When they constrict the pupil, less light is going in. And how they do this is by relaxing the radial muscles and contracting the circular muscles. If there's dim light, so it's very dark, you want more light to enter your eyes because you want to be able to see. You want to be able to figure out what's going on in the dark. In this case, your radial muscles are going to contract and your circular muscles are going to relax. And this causes your pupil to then dilate. So it works very similar to the distances where if it's closer, they work antagonistically where one contracts and one loosens. This is the same thing. One contracts, one relaxes. And it allows the changes of the pupil to become bigger or smaller, so dilate or constrict, depending on the light environment. And the reasoning behind this is because if there's too much light, you don't want to damage the eye, so you make the pupil smaller. And if there's very little light, you want the pupil to be bigger so that more light can go in so that you can see more clearly. Was that clear enough? Is everyone in triple science understanding that better? Which are radial muscles? I don't think you need to know where they are. I don't think they ever ask you that. So hopefully that was clear enough. Yeah, perfect. Yeah, okay, let's move on. Okay. Okay. Now, I want to give you guys, this is not on the eye. Actually, this is on reaction time, and I believe this is in both triple and combined sciences. So I want everyone to kind of have a look at this question. It's about reaction time, and I want to kind of look through this because planning and investigation question, I didn't talk about this in command words, but planning and investigation questions are very, very common in biology, and they're very, very useful because there's a very specific structure to how they want you to answer it. So I'll give you guys a minute to look at this question and I'll go through it. Okay, hopefully everyone has had a look at that question. The way you want to do plan and investigation questions, I know this topic might be a bit harder for combined sciences, but the reason I'm doing this is because plan and investigation questions come up in every single exam, and the structure is going to work for every single person exactly the same way. So let's go through it. This one is just talking about reaction time. Rather than me talking about the answer, I just want to talk about how you formulate the answer. With plan and investigation, the first thing you talk about is just basically describing the experiment you're doing. So in this case, probably the ruler experiment, like dropping a ruler and then catching it. I think some of you had done this before. And in this case, just describing what's happening in the experiment. Drop the ruler, hold it, and then you talk about the variables. You talk about what you need to control. Sorry, let's talk about independent variables. So what you need to change, what you're going to do with the experiment, who's taking the coffee, who's not, how much are you giving? Then it's the control variables. This is another section where they want you to get marks from. So what you would control, the amount of coffee you're giving them, the type of coffee you're giving them. So to answer the plan and investigation question, you would talk about the experiment. So you're describing the experiment. Then you're going to talk about the controlled variables. And the last mark is results. So it could either be analyzing the results or making sure the results are accurate. So things like repeating the experiment or writing a table to analyze these results. Anything along those lines will be fine, and write 2 to 3 points in each section. Write 2 to 3 points of describing, 2 to 3 controlled variables, and 2 to 3 points explaining the results and how you'd analyze it. So here is the mark scheme. So you can see here, the top part tells you describing the experiment, middle part talks about explaining the results and how you make them accurate, so doing it multiple times, comparing results. Final thing, control variables. I hope this is clear for everyone. This is just a method to answering exam questions.